Okay, so this is part two. So we're just going to continue going through proteins, and fairly straightforward in terms of the objectives for this. There's a little bit of sort of detail on the protein folding, but no. Hopefully this will be a bit more of a relaxing sort of revision video than the first half. So we're going to look now. Bear in mind, we now know the chemistry of amino acids. We know how the peptide bond is formed. We know what that looks like. We're now going to look at the levels of protein folding. And then we'll look at some examples of some proteins that you need to know, so fibrous and globular ones. So the first thing to note is that the first level, so number one, the first level of protein structure is called primary sequence or primary structure. Okay, very simply, this refers to when the protein is first made and we have the linear chain and actually the sequence, the order of amino acids that we have. Okay, so all the amino acids have different names, they've got different R groups, and the exact sequence of amino acids that we have that get joined together, and I'm just going to draw the amino acids as blobs here, connected by these bonds. So one here, one here, one here. These are peptide bonds, remember? So peptide bond. The sequence of amino acids that we have in the kind of linear chain before it does anything else is known as the primary structure. Okay, so this guy might be lysine, this one might be arginine, tryptophan, valine, you might have a glycine, glutamate here, and then maybe an asparagine here. So that particular order is the primary structure. Of course, I'm drawing the amino acids as blobs. They are, of course, the chemical molecules we came across last time, and they've got R groups, of course, sticking out, right? Don't forget, we've got different R groups. Some of them might be positive, some of them might be negative, etc. So fairly straightforward, I hope. The second level of protein folding is something called secondary structure. So I often represent secondary like that, but it's sort of called secondary structure. And the first thing that happens to our protein, our sort of primary amino acid sequence, is we get, just check we're still recording. Yes, we are. Is we get local folding now. And what I mean by local is it's very important. This means it's between amino acids. Amino acids, I abbreviate like that, between amino acids that are close together in the initial primary sequence. The important thing is this basically only happens because of the peptide bond, the intrinsic properties of the peptide bond. Okay, and I'm going to put this in a sort of bubble over here. It has nothing to do in terms of actually forming these bonds. It has nothing to do with the R groups. Okay, really important. Absolutely has nothing to do with the R groups. So what do I mean by this? Well, let's take a particular protein sequence. I'm not going to draw every amino acid in. I am going to draw the peptide bonds, however. So at the first end of the protein, of course, we have our carboxylic acid group that reacts with the amino group to form the peptide bond that looks like that. And of course, it continues that way and continues this way. And one thing I'm going to just point out now is the chemistry of the peptide bond. Not only did we say that it had a resonance structure, where the double bond moves to the carbon-nitrogen bond there, but also we've got a delta negative oxygen, which means that the oxygen is delta negative, the carbon slightly delta positive. The nitrogen here, delta negative, and that makes this hydrogen delta positive. So let's keep going. Let's add another amino acid in. So here we go back to the C terminus. Then we have the NH group, and that again bonds somewhere else. And you can imagine this keep going. Eventually, you can imagine that it kind of bends around like this, and we have this arrangement like that. Okay. And as it bends around, this particular amino acid down here becomes very close to this amino acid peptide bond up here. Okay, the peptide bonds. And hopefully what you appreciate is if I put my deltas in again, that we now have hydrogen bonding potential. Because remember, for hydrogen bonds to occur, we need a hydrogen that is covalently bound to an electronegative atom, in this case nitrogen. Remember, FON. And we need to have another electronegative atom, typically FON, fluorine, oxygen, nitrogen, nearby. Oh, look, we do. So what this means is we can actually form hydrogen bonds between amino acids that are close together in the primary sequence. Okay? So this can keep happening, right? We can imagine that this maybe bends around again, a few more amino acids get added, and eventually we get to an amino acid that lines up like this. And the same thing happens here, right? You've got delta negative here, delta positive there, and you have another hydrogen bond that forms. And you can imagine this keeps going. Okay, I'm not going to labor the point too much, but this could happen again here. Blah blah blah. Okay, so another hydrogen bond here, and this can keep going. And what this forms is a sort of coiled structure like this. Okay, whereby your hydrogen bonds exist between the peptide bonds, between the NH group of one peptide bond and the C double bond O of another, which I'm sort of showing like this, right? Like that. And these are hydrogen bonds. And because of this, we can form a helix, and we call this an alpha helix. And that's the first type of secondary structure that you need to know. Okay. And it's because we have the hydrogen bonds that form between the peptide bonds, between the sort of carbonyl linkage and the amide linkage, the NH group and the C double bond O group. And that's repeated. And that means we can, in a similar way, I guess, to sort of amylose forms a coil, this can as well. It's important to note that the R groups stick out the side of the structure. They do not participate in the formation of these hydrogen bonds. Okay, so the R groups of all the amino acids are sticking out of the structure like this. Okay, they stick on the outside of the alpha helix. The other point to note is, as you can see, I've drawn here, if you were to look at one peptide bond, so look at it like this, if we draw the deltas in, you can see that the extreme ends of the peptide bond, on this end here, we've got a delta negative, and on this end, this end down here, we've got a delta positive, this end delta negative. So the whole peptide bond has polarity. So it has a delta negative end and a delta positive end, right? This is a dipole. Okay, it's dipolar, it has a dipole here. And hopefully what you can sort of see from the way that I've drawn it here is that the peptide bonds line up so that they face the same direction. In other words, upwards, right? That's facing that way, like that. Okay, so the dipoles, if I were to draw them in, the big dipoles, like I just drew over here, poles face in the same direction. So the dipole for this peptide bond is facing this way, where we've got our delta positive here, delta negative here. The dipole for this one, oops, missed the hydrogen out, there we go. Dipole for this peptide bond, again, delta positive at the bottom, delta negative at the top. Same thing for this one, right? So the whole helix has an overall dipole. Okay, so the overall, that's a horrible line. Can't get rid of it now. There we go. The whole helix has a huge dipole essentially, where we are delta positive at the bottom here and delta negative at the top. Okay, so the whole alpha helix actually forms like a sort of dipole in its own right. That becomes important in upper sixth when you look at sort of ion channels and things. So if you want a synoptic link, two ion channels, and how we can kind of gate them, right? Open and close ion channels, and we'll talk about that when you do the nervous system. So in neurons specifically. So that's just a nice sort of link to upper sixth. It comes in much later on, but for now, just appreciate that we have a overall dipole. It's like a super dipole, right? The whole alpha helix has a dipole. So that's the first one. We don't just have to form a helix, though. There is a second option available to us. I mean, there are more actually, but A level you need to know too. And this is called a beta sheet. Okay, it's sometimes called a beta pleated sheet. So in here, sometimes we put the word pleated because it has a pleated structure. And this is where we don't form a helix, right? And actually, hopefully what you appreciate here is that we'll have some amino acids that, are here, for example, like this, where the peptide bonds that are shown here are not used to form the helix. However, the beta sheet uses every single peptide bond. So each sort of peptide linkage is used up in bonding. Okay. And so we don't form a helix. In this case, what we actually do is form a sheet. So it kind of, looks like this, right? So the kind of chain primary sequence folds up, okay? And essentially, in fact, I'll draw it laterally, might be easier. You get this. So if I draw the peptide bonds in, I'll draw a few amino acids, peptide bonds. Don't forget, I'm not drawing like the alpha carbon and all this other stuff. I'm just focusing on the peptide bonds. But you can imagine that then bends round in a sort of more flat structure. And we have this. And it can then bend around again. Okay, but I'll just keep it at this for now. Don't forget we've got deltas. So I'll just do one example here, delta negative, delta positive, and our hydrogen bond, of course, between them like that. And this can continue to go around, and it kind of sort of loops around in a sort of flat structure like this, okay? And every single peptide bond linkage is involved. And that's called a beta sheet or a beta pleated sheet. Okay, so it's a fairly flat structure. It's not a coiled helix. It's a fairly flat structure. Okay, and we can have two flavors of this. You can either have it, if I just draw a bit like that, is facing in an opposite anti-parallel direction. And that's kind of what I've shown in my diagram down there at the bottom, where we have our hydrogen bonds between the sheets like that. And this is an anti-parallel, okay? Anti-parallel beta sheet. But you can have it whereby you have a parallel beta sheet. So it's hard to draw. I've got a picture of it later. But imagine we've got a particular beta strand, so an individual strand within a sheet is called a strand. It kind of then loops over itself, and then sort of forms a strand going that way again, and sort of does the same thing. And I've got a better view of it. I've got a picture that I'll show you in a bit. But these strands are all parallel now. So not only do we have anti-parallel, we also have parallel beta sheets. Okay. That this really makes a difference is the strength of these interactions. Okay. If you are a anti-parallel beta sheet, hopefully what you can appreciate here is because we're going in this direction on this strand and we bend round and then go in this direction, the hydrogen bonds that I've drawn here line up perfectly straight with each other. So the double bond oxygen lines up nice and sort of parallel to and kind of straight, sort of head-on alignment with the NH group. Okay, that means these are stronger because they can get closer together. Okay, so you get a stronger force of attraction, electrostatic force of attraction. If you're a parallel beta sheet, the hydrogen bonds here actually form, because of the constraints put on sort of shape and structure with regards to the peptide bonds, they actually form, and I'm emphasizing the point, like an angle. And what that means is they are further away from each other, and so the hydrogen bonds are weaker. Okay, you have a weaker hydrogen bond, a weaker electrostatic force of attraction, and that is because you don't have direct alignment between the peptide bond sort of segments that are actually doing it. Okay, that's pretty much all we need to say with regards to that. So we can form either of these two structures, either an alpha helix or a beta pleated sheet, beta sheet. I'm just going to show you the structures of those now in sort of diagrammatic form. I hope that's going to be more useful to you. So if I scroll down, I had them here. Here we go. So this is what the alpha helix looks like, just in kind of a better pictorial form. And you can see we've got our intrachain, meaning within a chain hydrogen bonds. Not every single amino acid peptide bond is used up. And actually it forms if you're interested, a right-handed helix. So you can use your fingers and your thumbs to work out which way you're going. And if you curl up this way, that means you're a right-handed helix. Don't worry about that. But you can see that here you've got your C double bond O, which is delta negative, hydrogen bonding to your NH group, which is delta positive. Same thing here, right? Delta negative here, delta positive here. So we've got our overall huge dipole, don't forget, again, delta negative, delta positive, going this direction, where we're delta positive up here and delta negative down here. Okie dokie. The only thing I just want to point out with this, I was not going to show you this, but I think I will, is just to say that different amino acids have different propensities or different sort of abilities or preferences, I guess, to form either an alpha helix or a beta sheet. Okay, and you don't need to know this, but something like tyrosine, for example, down here in yellow, is majority found in a beta sheet rather than alpha helix, right? Whereas something like glutamate up here is often found in an alpha helix. If you're interested in why that is, then speak to me. Okay, right. I'm going to stop that there in terms of secondary structure. To tertiary structure. And it's important to note, okay, actually, before I do that, about the local folding of secondary structures, okay? So our secondary structure is local folding. What that means is in my primary sequence of amino acids, right, where my amino acids are joined together via peptide bonds, it might be that the first bit of the protein forms an alpha helix with these amino acids. We then maybe go into a beta sheet, and then maybe this bit forms an alpha helix as well. Okay. And what that means is, or my point is that only the local bits of the protein, right, bits that are close together, amino acids close together, form, in this case, you've got an alpha helix. Here, you've got a beta sheet. And here you've got another alpha helix, right? But, one end of you have to be close together, okay? So it's on quite a small scale. Tertiary structure is interactions between amino acids. So amino acids interact with each other now that are far or can be far apart. And this is the important point. We absolutely depend on R groups for this. Okay, this is where your R groups come in. And actually, it's the R groups now that determine, and this is what tertiary structure is, the overall, so tertiary structure is the overall three-dimensional shape of the protein. Okay, and that is determined by the order of R groups that you have. Okay, so hopefully what you appreciate is, let's imagine I've got this long chain of amino acids, and I've got one R group here that is positively charged, and I've got an R group way over here that's negatively charged. Okay, I'm going to put my secondary structures in now. So not only do I have that, but let's say this positive charge is within an alpha helix. And then we go into maybe lots of beta sheets, maybe something disordered bit there, more of a beta sheet there. And let's imagine in this example that there's another alpha helix here that contains the negative charge. Maybe that's over here. And that contains the negative charge. And hopefully what you can appreciate is that these two are going to want to interact with each other, right? They're going to want to bond via an ionic interaction. And that's going to bring, as I've kind of alluded to here, this end of the protein into close proximity with this end. So you'll end up with it sort of looking like this, right? You've got your alpha helix here with your positive amino acid R group. You've then maybe got all your beta sheets. Oops, that's really bad, beta sheets. Right, beta sheets. And you then have your other alpha helix with your negative R group. And they're interacting via an ionic bond. Okay? And the reason that your protein has now folded into this shape is to get these two groups close together. Okay? And that's just one example of how these R groups could interact. Okay, so go back, look at your R groups and see which ones, this one might have, for example, an NH3 plus group. This one might have your COO minus group, for example. We gave an example of that earlier. But of course, we don't have to just rely on ionic bonds. All the other types of bonds can exist and can dictate how this protein is going to fold up. So that's the ionic bonds. And we're now just going to talk through other interactions that can happen, right? So we can have ionic bonds between our groups. We've looked at that. We can also have something called a disulfide bond or a disulfide bridge. Okay. Now this is a covalent interaction. So I'm going to focus a little bit on this now. It's a covalent bond. Okay. And this is where the amino acid cysteine comes in. Okay. Actually, I'll write that clearer. And if we scroll all the way back up to our amino acid R groups, and we look at cysteine here, I told you that we needed to remember it because it's got a thiol group. Okay, it's got this SH group on it. Okay. So why is that important? Well, in our chain, our primary sequence chain, right, where blah, 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 over here we might have an R group that's cysteine, and the R group contains our thiol group, SH. And we might have one in this beta sheet. And they can interact with each other, right? Two cysteines. And essentially, if I just box off cysteine as a box and just show you that the SH group is here, and I'll do it again here. If we've got two cysteine amino acids and they're close together, we can actually react so that these two hydrogens get liberated. And this produces two hydrogen atoms, which then gets split into two protons and two electrons, right? Each hydrogen atom, don't forget, is made up of a proton and an electron. So if we've got two hydrogen atoms, we've got two of each, and they get split up like that. And the result is we then have a bond between this cysteine on the left and the cysteine on the right here via two sulfur groups, two sulfur bridges. So this is called a disulfide bond. Okay, and it's a type of covalent bond that can happen. Okay, and by the way, we have lost electrons here. So this is an example of an oxidation reaction. And it's important to note that it is an equilibrium, so we can reduce this and go back to the sort of individual cysteines, but I'll leave that there. The key point is this is a covalent bond. It's a strong covalent bond. And this helps the proteins resist being denatured. Okay, so it helps resist denaturation. Okay. So if you've got lots of cysteines in your primary amino acid sequence, you're more likely to have a more stable structure. Okay. Point that out. I think I must have given you a picture for that. I can't remember now. Oh, I forgot the beta sheets. Here we go. Sorry, just going back to beta sheets. This is basically what I'm showing you. I forgot to show you this. Obviously, these are parallel beta sheets, right? Because each beta strand is going in the same direction. And that's because we kind of loop this little bit out between each strand. Or they can just fold up right next to each other and be anti-parallel, as you can see here. Okay. And we call it a beta pleated sheet because actually it looks like this in sort of 3 dimensions, right? It has this kind of pleated, zigzaggy structure. It's quite flat, but it has a zigzag structure. It's called pleated. And this is just showing your hydrogen bonds here for a hydrogen bonds are at angles to each other, therefore they are weaker. Whereas the anti-parallel, so this arrangement, they are absolutely sort of head on to each other and much stronger because they can get closer together. Okay, you can hopefully see this, 4 dashed lines here, much closer than however many these are. 6 dashed lines over here. Sorry, my OneNote's being weird. Right, I'm hoping I've got the cysteine thing. Doesn't look. Oh, I have. Here we go. So here we go. Two cysteine residues can form a disulfide bond, as shown here. Oops, that's horrible. I'll leave it like that. We can oxidize them to join them together and liberate 2 electrons. You can go back via reduction, but we're not going to worry too much about that. Okay. Just a nota bene. Cysteine here is the individual amino acid. When we've got 2 together, 2 cysteines with an E, we call the whole thing a cysteine with a I, just to confuse things. But this is an example of the primary sequence of insulin, okay, polypeptide. And you can see here that we've got disulfide bonds. We've got disulfide bonds within a primary sequence, okay, within one of the chains. That's an intramolecular bond, disulfide bridge. And we've got two of them here that actually connect two completely separate proteins together. Okay, so that's an intermolecular disulfide bond. Again, if you're unsure about that, do hit me up with an email or speak to me in class. Gosh, we're covering a lot of material here, but we're nearly there. I'm just going to check my battery on my Surface device. I'm just going to plug it in because I think we're about to die. Okay. So that's our cysteines and our disulfide bonds. So that's the other type of bond aside from ionic bonds. If your R groups, don't forget, have in this case 2 thiol groups, because you've got 2 cysteine residues, 2 cysteine amino acids there. We can then obviously do this, where it's going to do, what is it going to do? It's going to do this, and then this. And our disulfide bonds are going to be like that. Okay, and that's a covalent linkage. And again, that's going to help determine your tertiary structure, okay, your overall specific 3-dimensional shape that your protein is going to fold up into, okay, as opposed to the secondary structure, which is a sort of much more zoomed in local region folding, right? So, you know, this is secondary structure, this is secondary structure. Multiple secondary structures exist in the same protein, but they're kind of in their own little regions. And actually, you know, in large scale interactions between amino acid R groups is tertiary structure, and that actually gives the protein its overall shape. And of course, you can see here, look, you know, here we've got an ionic bond. Let's imagine we've got a disulfide bond maybe somewhere else that's helping with this. And hopefully what you can appreciate is this is starting to look a bit like an enzyme, right? This could be the active site in yellow here. The active site, look. Okay, so hopefully that makes sense. Right, and we are nearly there. So that's the disulfide bonds, ionic bonds. There's a couple more to talk about. The next one, are hydrogen bonds. So I think this is number 3. The next type of bond that can stabilize this structure is a hydrogen bond. And we've talked about this before. I'm not going to label the point, apart from to say that, you know, imagine we've got our usual situation like this, where we've got lots of secondary structural bits. And, you know, imagine we've got 2 residues here. One R group here contains a hydrogen bond donor, and one contains a hydrogen bond acceptor. Okay, so maybe something like an NH2 group, something like that. And here we've got a C double bond O OH group, something like that. And hopefully what you can appreciate, if they're both protonated, we can have a hydrogen bond here between these two, okay? But just to link back to what we said before, whether this is protonated or not, or whether this is charged, of course, depends absolutely on the pKa values that they both have and what pH we're at. Okay, so there you go. That's that. Just know that hydrogen bonds can help stabilize the structure. The third thing is something called a hydrophobic interaction. Fourth thing rather is a hydrophobic interaction. And this is mainly just to say that you can have R groups that are hydrophobic, that hate water, and they're going to cluster themselves together on the inside of the protein. So I'll draw it out first. I think that makes sense. So again, imagine I've got an alpha helix. Maybe I've got, you know, beta sheets here, another alpha helix here. If here I've got R groups, don't forget we're in a cell. There's loads of water, right, hanging around. I'm not going to draw all of them in, but there's lots of water hanging around here. So when this folds up, not only have you got ionic bonds, hydrogen bonds, and disulfide bonds that can form, these amino acids here are going to want to interact with each other because they're all hydrophobic they are not going to want to interact with water. So what's going to happen is they're going to cluster together, okay, on the interior of the protein, okay? So I'll kind of show that like that, where those R groups are going to cluster and interact with each other very closely in the inside of the protein and try and get as far away as, from water as possible. So don't worry too much about that one, but just be aware that it's a thing. So I'm just going to show you here. This is kind of what I've been talking through. So we've got hydrogen bonds on the left here. So if you had an R group, because it shows the amino acids, which is quite nice, but the R group here again would be this whole group. And if you've got a double bond oxygen here and it's near another hydrogen attached to something delta negative on this R group here, you can have a hydrogen bond between amino acids. So if you had a glutamate nearby to a serine, you'd have a hydrogen bond. Something like aspartate and lysine, these are charged amino acids. So you've got a positive charge and a negative charge. Okay, aspartic acid is negatively charged. Lysine is positively charged. You can have an ionic bond. Remember, this depends on the pH, right? Because this group here will have a pKa value associated with it, as will this group. So whether they are actually positively and negatively charged like this depends on the pH, right? So just be wary of that. If you change the pH, you might knock out this positive charge here and stop the ionic bond from happening. So, and that will destabilize protein shape and therefore function. Again, speak to me if you're unclear about that, but I don't want to labor that point anymore. And this is just showing hydrophobicity. So if you've got groups here that are pretty hydrophobic, so leucine and isoleucine, pretty hydrophobic, they will interact with each other. They will cluster together, typically on the interior of the protein, to get away from water. So that kind of helps fold proteins as well. Okay. The final stage of protein folding, before we look at two examples, is quaternary structure. And I'm not going to spend too much time on this, apart from to say that this is when we have more than one polypeptide chain that interact with each other. Okay, they can interact with each other by all of the forces that we've talked about for tertiary, right? So they can do this via ionic bonding. Horrible writing, sorry. I've been writing a lot today, so my handwriting is getting worse. Hydrogen bonds. We can have hydrophobicity interactions, and of course covalent linkage. We can have our disulfide bond as well. All of these things can help with allowing one sort of protein blob here like this interact with another protein, completely separate polypeptide chain, right, that will bond to it. Okay. And there are many examples of this that you'll come across, but it's in the notes, so do read through the notes. But I do want to point out that this happens with the majority of proteins. Okay, so most proteins do not simply exist as one blob, like one tertiary structural blob. You actually have multiple tertiary structural elements that come together to form a big protein complex, okay, which is quaternary structure, more than one polypeptide chain, individual polypeptide chain. I'm not going to sort of say much else on that at this stage, because you'll come across lots of examples of this later. I'll just give you one quick example, which will come up in the DNA topic we're doing next, which is that if you look at how DNA is replicated, it is done in part by breaking the two strands of the DNA duplex apart, and that is done by this guy here. Oops, it's supposed to be yellow. which I've zoomed into here and given you the structure of, and it's called a DNA helicase. Okay, in bacteria, it's called DNAB. In humans, it's called MCM. And hopefully you can see here, it's kind of shown in that picture there. It's made of lots of blobs. Each colored element here is a tertiary structural protein. It's an individual polypeptide chain. They link up with each other by bonding in, R groups bonding to each other, right? Disulfide bonds, ionic bonds, all that kind of stuff. And you can see here there's 6 of them. And what's nice about this picture is you can see alpha helices, look. And you can see maybe like a beta sheet structure, maybe there, right? Or here. So it kind of shows that you can see these structural elements. And essentially this forms, if I was going to cartoon draw this, it would look like this, where you have individual protein chains that are interacting with each other via all these bonds, right? Maybe some of them are covalent linkages, disulfide linkages. Some of them are ionic bonds, hydrogen bonds, etc. And they link together like that, and they can, perform a function, right? So this is a big complex, lots of individual tertiary structural elements together. Okay, so this is a good example of quaternary structure. But most enzymes, most proteins actually exist in quaternary structure. Okay. The key thing is to know the definition. It's more than one polypeptide chain that interact with each other. Just a point, actually, if I just go back to this, just in case I forget to mention it at any other stage during our lessons. When you have tertiary structural elements linked together like this, each tertiary structural blob, so this one here, for example, is called a subunit. Okay, so when you have your quaternary structure, each one of these colored things here, each one of the tertiary structural elements is called a subunit. So in this case, we've got 6 subunits joining together. Okay, enough said on that. Right, so going back up to where I wanted to get to, we've done PHPKA. Here we go. So that's levels of organization done. So primary sequence, secondary, tertiary, and quaternary. And we're finally now going to look at 2 proteins very quickly because you don't need to know much about them. But we're going to look at fibrous and globular proteins because we've drawn them as blobs so far, but they're not always. Right. 2 types of protein that you need to know that can form. We can have fibrous proteins, and these are structural. That's their job in the cell. They help with structure. Okay, so they tend to be like sort of long chains. Okay, that I don't know why I'm doing exclamation marks, but they tend to be structural and they're chains. The example you need to know is collagen. Okay. And if you're interested, about 1/3 of it is made up of glycine amino acid R groups. You can look that up in your own time as to what those R groups are. It's a very simple R group. Actually, I may as well just tell you, it's just a hydrogen atom. And hopefully you can appreciate that that allows these chains to get close together. Ring. The next thing is to say that the primary structure, the amino acids, fold up into a secondary structure. And the tertiary, this is the important bit, the tertiary structure is a chain. Okay, it's like a helix. Okay. Not to be confused with the secondary structure, which may contain helices as well. So if you imagine, right, we've got, actually, I may as well draw the whole thing, right? It forms a helix like this, right? But that's the tertiary structure. The secondary structure, which I will do in a different color so you can see it, if we zoom in, we'll have an alpha helix here, right? You might have a bit of a gap and then another alpha helix, right? You might have a gap and another alpha helix. You might even have a beta sheet perhaps here, or another alpha helix here. But if you zoom out, the overall tertiary structure, which don't forget is stabilized by not the peptide bond, but interactions between an R group here that might be positively charged, let's say, and an R group here that's negatively charged, right? You might even have a disulfide bond here. This is now the tertiary structure, right? So we're zooming out. This is the big picture here. And that's just one protein. That's just one chain. That's a subunit, essentially. But we have quaternary structure. And the quaternary structure is we have 3 helical chains that interact with each other. So this is 1 helix, and we're going to have 3 of these. So let's put one here like this. Let's put one here like this. This is a horrible diagram, and I apologize. There's a much better picture coming up, and there's another one like that. And they can interact with each other by using, obviously, ionic bonds, hydrogen bonds, blah, blah, blah. Okay. And the key thing is that this makes it very strong. You can have lots of interactions between them. And structurally, therefore, it's good for structure, right? So collagen is a really important structural protein. Okay, that's pretty much all you need to know. I don't know if I have a picture to show you on that. Annoyingly not. Okay. I mean, you can look that up in your own time. It's in the notes, actually. It's not a very... Draw it in pen. It might be better to show it like this. That's one. That's another. And that's another, right? They kind of interact with each other and they wrap around each other in like a coil structure. So hope that makes sense. You don't need to know a huge amount about it, to be honest. That's basically the extent of it. But it's good for structure, right? They're strong, okay? They tend to be chains. The other type of protein are globular proteins, and these have metabolic functions. They're not structural. Okay, their metabolic function. What that means is they tend to be something like an enzyme, or there could be sort of hemoglobin or antibodies, that kind of thing. Okay, we'll come across structures of these later. Essentially, the tertiary structure is a big blob, and you may have quaternary structure where we have lots of blobs coming together. In the case of hemoglobin, you've got 4 blobs coming together. You've got 2 alpha chains, 2 beta chains. They interact with each other to form a quaternary structure that combines. Okay, I'm rushing this deliberately because you don't need to know much about that, and we'll come across hemoglobin as a big case study next term. Just know that globular proteins are enzymes. They have catalytic functions generally, or in the case of like hemoglobin, it's sort of a general metabolic function of binding oxygen, of course, to all of these subunits. Okay, final thing is how we test for proteins, right? The biochemical test for proteins. Very simply, take a test tube with your sample, and you're going to add an equal volume of sodium hydroxide solution. We then add a few drops of typically 0.05% solution of copper 2 sulfate. Copper 2 sulfate. And this we actually call a special name. It's not Benedict's reagent, that's in carbohydrates. It's slightly different. It's called biuret reagent. That's what you can call it in the exam. Biuret reagent. And if it's a positive result, the change we have is we go from blue to lilac. And obviously, if it's a negative result, we remain light blue. And you don't need to know how that works, and I'm not going to bore you with the details. So that's that. Again, any questions, email me. That's proteins done. Well done. So please do make the notes in your own time. As I said, I will check through your notes once you've done that. Any questions, get back to me. Always happy to meet either in person or on Teams. Just ping me an email or speak to me in the lesson. Cool. Thanks very much. Well done for getting through it. Long session for proteins, but hopefully nonetheless useful to you, at least if not now, for revision purposes.